Alright folks, so this is chapter 27, fluid electrolyte and acid-base balance, and this first part covers 27-1 and 27-2. So first of all, our body is divided into fluid compartments, and there's movement between all these compartments from one compartment to another. So if you look at the total body water, and of course this is variable. This is by no means you or the person sitting next to you, but it's approximate. You'll be around these numbers. You have about 40 liters of water in your body, and it's about 60% of your body weight. Okay, and all of this body water exists in two main compartments. Those two main compartments are the intracellular fluid, that's your cytoplasm, that's in your cells, and your extracellular fluid, and that is all the water not in your cells. Okay, now the extracellular fluid can be divided up further. Most of your water is in your intracellular fluid. Now, some books call this the 2/3, 1/3 rule. Okay, they say this is 2/3 and this is 1/3. You can see that the numbers aren't exactly 2/3, 1/3 here. Uh, but it's close. It's not far. And again, these are approximations anyway. Just so you know, these are approximations. Your intracellular fluid is most of your water. All right. Of the 40 liters, 25 liters of it is in your cells. And of that 40 liters, 15 liters of it is in your extracellular fluid. Now, what is the extracellular fluid composed of? Because I said that can be divided further. And indeed, it can be divided further. It can be divided up into interstitial fluid. That's the fluid between cells. And it can be divided up into the fluid in your plasma, the liquid portion of your blood. So, and by the way, you're probably saying to yourself, well, what about lymph? And you'd be right. It can actually be divided even further. The extracellular fluid can be divided even further because I'm not accounting for the lymph fluid that returns to your plasma, but for a snapshot of time, there's fluid in your lymph system. So you could have lymph in here and add that to it, and of course you would not be wrong. So this picture is just showing an approximation of what's going on. All right. So that's the compartments of your body water. These are the electrolytes that exist inside these different compartments. And in your interest, so there are a couple of things you have to memorize here. And let me just come right out with it. You probably know this already because we started out the semester talking about this. Here's what we started out the semester saying. Sodium is a major extracellular cation. And look at, in the two compartments that I'm labeling right here are extracellular. Sodium is a major extracellular cation. Chloride is a major extracellular anion. All right. Potassium is a major intracellular cation. And you can see intracellular. Phosphate is a major intracellular anion. Okay, so you know those already. You know the sodium potassium chloride story. You pretty much know those. You also know that proteins are a major intracellular anion. Because the reason why the inside of the cell is negatively charged relative to the outside of the cell is these proteins are negatively charged. That's not to say there aren't proteins outside the cell. Look at, there are. But these proteins right here, and the main protein in your plasma is albumin. There's other ones like fibrinogen and stuff, but the main one's albumin. They're not a big part of the extracellular compartment. So you can see this here. Let me point out, if there's anything else I want to point out. Bicarb can be in both. So bicarb, you can see about equal, not quite equal. It can be in both extracellular and intracellular. It moves back and forth as a buffering system. So we'll talk about the buffering system later. And just because sodium's major extracellular cation doesn't mean it's not found in the cell. It is, but very little of it. Just because potassium is a major intracellular cation doesn't mean it's not found outside the cell. It is, but just a little of it. So you can see all of these right here. Now look at this. This is kind of important. This little green sliver is calcium. We keep intracellular calcium at a very low concentration. However, it would surprise you. Well, let me say it like this. Even this little bit that we have here, it's not allowed to float around the cytoplasm or the nucleoplasm. Because if it were, it would precipitate out DNA. DNA is negatively charged, and calcium is positively charged. It would bind to those phosphate groups of DNA and precipitate it out. So even the little bit of calcium that we do allow inside the cell, it's compartmentalized inside the cell. It's stored in the smooth endoplasmic reticulum. So yeah, there's a little bit of calcium inside the cell. And as you know now from anatomy and physiology courses, that transiently calcium is allowed in the cytoplasm because it controls certain physiological events. Like it could be a second messenger when it's complex with calmodulin. It's involved in muscle contraction, etc. But we do compartmentalize it in the smooth endoplasmic reticulum in the cell, and we expend energy to do that. All right, so you can see all of these electrolytes, the anions and the cations, and in what compartment they exist. This picture is not in your book, but it just says to you, basically, whatever you take in a day, you have to output in a day. So if you take in 2,500 mils, which is 2.5 liters, then you better get rid of 2.5 liters in a day. All right, I'm not saying that you don't change it. I'm not saying that you don't utilize it in the meantime. But whatever you have as intake, you better have as output. Otherwise, you're going to blow it up with fluid or shrivel down like a raisin. So your intake and output must be equal. And as a healthcare worker, you might measure I's and O's. I think it's called intake and output. Remember, I'm not a nurse or anything, and I'm a medical technologist. I've never measured I's and O's. I just know they do it. And the lab does do a lot of testing that gives a snapshot of time of what the hydration status of the patient is. So we do indeed do this. And of course, the lab tests urine and other body fluids. The lab tests feces. So the lab is definitely involved in testing some of these things that you're seeing as output here. All right, so that's intake and output, and they have to be equal. This is showing you different systems that are involved in the balancing act here. All right, so you have the cells doing metabolism. That's what cells do. They metabolize, and when they metabolize, they create waste products. All right, CO2 and nitrogenous waste products as one example. They require nutrients to do this metabolism. You can see here. And here's another nutrient, but it's oxygen. That's the terminal electron acceptor in the electron transport chain. So how are we moving between these compartments? Because here's the intracellular compartment, and both of these are the extracellular compartment. And how do we move? Well, we get some things into our body through our lungs. We get some things in our body through the GI tract. We get rid of some things out of our body via the kidneys. So you can see that all these are interrelated and just moving things back and forth between these compartments. Same picture from before. Before I was just showing you the compartments. Now I'm showing you, well, I'm actually still just showing you the compartments. And again, these are approximations. This percent body weight of your total body water differs between men and women because men have a higher lean body mass than women on average. So this is just an approximation. The big thing I want you to know is fluid between these compartments moves. It moves between the plasma and the interstitial fluid and between the interstitial fluid and the cells. It's always moving. Well, It is always moving, like I just said, but it remains relatively constant. So homeostasis is maintained in these compartments. Okay, here's the basic concept of all of this. All of your homeostatic mechanisms, every one of them responds to something in the extracellular fluid. There is no receptor that points to the inside of the cell. Now, receptors are on the cell membrane and point to the outside of the cell. Receptors are detecting things in the interstitial fluid. There are receptors on your endothelial layer of your bloodstream, of your vasculature, your blood vessels, that detect plasma. So, sure, the receptors are part of the cell, but they don't point inward. Let me go back and just show you what I'm talking about here. There are no receptors pointing inward saying, how salty am I? But there indeed are receptors pointing outward saying, how salty am I? So we have receptors monitoring extracellular fluid, not intracellular fluid. So that's one of the major concepts here, is our homeostatic mechanisms are responding to changes in extracellular fluid, not intracellular fluid. However, when we respond to changes in extracellular fluid, it affects the intracellular fluid because there's always fluid moving between those compartments. All right, number 2, no receptors directly monitor fluid or electrolyte balance. Now, I got to be careful how I say this because we certainly monitor osmotic concentration, which is, kind of electrolyte balance. But we monitor just that, osmotic concentration. If you're supposed to have a certain number of sodium ions, I don't monitor that raw number of sodium ions in my body. I say, you know what? I should approximately be about 140 millimoles per liter of sodium, and that's a concentration. But realize I could have a drop in sodium and a drop in my water and still remain 140 millimoles per liter. You see, I could, it might be 130 millimoles, but as long as I'm not measuring it in a whole liter and I drop my liter appropriately, the concentration is still 140 millimoles per liter. So I don't directly monitor the number of sodiums. I don't directly monitor 40 liters of water. I don't do that. But I do monitor osmotic concentration of my fluid. I indeed do that. And I monitor pressures. Blood pressure is one thing that my body monitors and responds to. And the blood pressure is directly related to how much fluid is in my body. All right. Water never moves by active transport. Water follows salt. So if I want water to move, I change the tonicity. If I want water to move inside my cell, I change the tonicity of my cell. If I want water to move outside my cell, I change the tonicity of the interstitial fluid. Water follows the salt or solute. It's not always salt, it could be a solute. And the last huge concept is if your dietary gains exceed your losses, you have a rise in your water and electrolyte concentration. If dietary gains, if environmental losses exceed your dietary gains, your electrolyte and water electrolyte content goes down. It's as simple as that. What you take in must balance what you get rid of, or you have a rise or a drop in these things. So I know they seem like easy concepts and pretty self-explanatory, but we'll come back to them as we talk about all of this. All right, this is another way of looking at what's in your different compartments. You could look at, in your intracellular fluid, just water. And, you could say, okay, I got 20, however many liters that is, 20, whatever. That's how much water is intracellularly, and that's how much water is extracellularly. And I have interstitial fluid, plasma, and other. Oh, here's where I say other, like lymph. The first picture I showed you didn't have that lymph in there. So you can see that here. Another way of looking at it is a pie chart. I think one is probably female. Yep. Look at this is the difference. That's why I'm saying that these are approximate numbers. The difference is you can see that the percentages are different between males and females. Like my intracellular fluid in males is 33%, but it's 27% in females. So 33% in males and 27% in females. So these percentages are different, and that's only because the lean body mass in males is higher than the lean body mass in females, approximately, on average. So this is showing you the two. Please don't memorize. Well, I kind of want you to know the gist of it, though. Yeah, so I don't want you to memorize these pie charts by any means, and you can see that they're different. I would like you to know this right here. So this is an approximation, of course. It's not differing men and women, and it certainly is an approximation. But this would be good if you knew this. And when I say know this, that there's 40 liters of water in our body. 25 of it is inside our cells, and 15 of it is outside our cells. All right, so 25 and 15 of the stuff that's outside our cells. Our plasma is really very little. You already know there's 3 liters of plasma because we did that in Bio 203. So of that 15 liters that's outside our cells, 12 of it's interstitial. And plasma is about 3 liters. So that would be good to know. Please don't memorize percent body weights. And the reason I say that is because that changes whether you're a man or a woman. That's what it changes. It's slightly different. But total body water 40 liters, intracellular 25, extracellular 15. That'd be good to know. It'll give you a good place to talk from when you're discussing these things. You can see solid components in our compartments: proteins, lipids, carbohydrates. The nucleic acids, of course, are solid components, but they're inside the nucleus of the cell. You can see we have minerals. Now, you're going to say, wow, minerals is up there. We got more minerals than carbohydrates. Well, it's by kilogram. Minerals are heavy. You're talking about zinc and iron and selenium and magnesium. These are heavy. So, yeah, we're doing this by kilogram, and that's why minerals look so high. This picture we already saw when I just introduced you to electrolytes. Electrolytes are charged atoms or molecules, and you can see the intracellular electrolytes and the extracellular electrolytes. Again, the major concept is potassium is the major intracellular cation, phosphate's the major intracellular anion, sodium's the major extracellular cation, chloride's the major extracellular anion, bicarbs found in both compartments, intracellular and extracellular. So you can see these. All right, the overview of regulation. All right. So first of all, we detect extracellular osmolarity or osmolality. First of all, what's the heck's the difference? Well, first of all, osmolarity is osmoles per liter. Osmolality is osmoles per kilogram. Lality. You're going to say, what the heck's an osmole? An osmole equals a mole of osmotically active substance. And that means it has to be dissolved in my fluid. If you have a glass of water and you put salt in it, it dissolves in there. It's osmotically active. If you have a glass of water and you put a brick in there, it's not osmotically active. It's not dissolved in the water. So everything that's dissolved in our plasma or dissolved inside our cellular water or dissolved in our interstitial fluid, it's all osmotically active. So we can measure the osmolality of these things. And the osmolality is osmoles per kilogram. We have receptors that detect the extracellular fluid osmolality. All right, let's say we have high osmolality. We are salty. We are salty. All right. Osmoreceptors in the hypothalamus detect this. Meanwhile, because we're so salty, we have decreased saliva and a dry mouth. All right. Osmoreceptors in the hypothalamus will detect this. They activate the thirst center. They say, hey, drink. We take a drink. The water hits water receptors in our throat and tells us we just drank and we become less thirsty. All right. In addition, the water we swallow, we absorb in the GI tract, and this decreases our plasma osmolality. We just added water to a salty solution and we decreased the saltiness. So it brings us back to a normal osmolality. Now, if I'm too salty, I probably don't have enough plasma volume, blood volume. Okay, so why is this? I mean, I guess I could be too salty and be hydrated if I'm eating a lot of potato chips or something like that. But a lot of times I'm too salty because I'm dehydrated. I've lost water, but I haven't lost salt. So I need to increase my plasma volume. And I probably have low blood pressure if I have low plasma volume. And likely I do have low blood pressure. So what happens now? Well, the granular cells of the juxtaglomerular apparatus of the kidneys detect this low blood pressure and they secrete renin. The renin converts angiotensinogen to angiotensin. Ultimately, it leads to high angiotensin 2, which makes me thirsty. And it does all the other stuff that you know angiotensin 2 does. It goes to the aldosterone. It does vasoconstriction. It does the ADH. So see, the angiotensin 2 does all of that stuff, and I retain more water. So yeah, the same story you just learned in the last chapter is going to be part of our fluid and electrolyte balance. Okay, now look at this. I have increased osmolality due to increased sodium concentration. My osmoreceptors in my hypothalamus detect this. They send a signal to my posterior pituitary and say, release antidiuretic hormone. And I release antidiuretic hormone and it goes to the kidneys and it says, it puts the aquaporins in the distal convoluted tubules and the collecting ducts of my cells. I reabsorb more water and I decrease my osmolality. And I make smaller volumes of concentrated urine, which we're calling scant urine. So it's another thing that happens. Okay, now you have learned thus far that angiotensin 2 leads to increased aldosterone secretion, and I did not lie to you. However, I didn't tell you the whole story. Aldosterone also responds to two other signals. Aldosterone responds to a high potassium or a low pH, which is, by the way, a high hydrogen. All right? Three other things. It also responds to a low sodium. Now think what aldosterone does. Aldosterone goes to the kidneys and it says increased sodium resorption. Now remember that in the kidneys, here's my urine, and it's really filtrate, but I'm saying urine. And here's my cell. And here's my blood. Now remember something. If sodium is coming out of the urine into my cell, and then sodium is coming out of my cell and into my blood, I'm also losing potassium by countertransport. So when I reabsorb sodium, I secrete potassium. And something I told you before, that hydrogen ions, protons, can replace the potassium in that countertransport. It can be hydrogen ions. So think of this. If I have low sodium and aldosterone makes me increase sodium reabsorption, that's going to also make me increase potassium secretion. Likewise, if I have high potassium, I will increase potassium secretion and concomitantly increase sodium reabsorption. Likewise, if I have a low pH or high protons, the protons can replace the potassium here, and I'll reabsorb sodium and secrete protons. And it gives me my homeostatic control of sodium, potassium, and pH. And that's from aldosterone. So aldosterone doesn't only respond to angiotensin 2. It also responds to sodium, potassium, and hydrogen ions in the blood. Okay, that's aldosterone. Atrial natriuretic peptide. Atrial natriuretic peptide. Some people call it atrial natriuretic factor. This does the opposite of angiotensin 2. That's what this does. All right. What happens is this. I have high blood pressure. So this is high blood pressure. That's what it starts with. By the way, I also have a brain natriuretic peptide. Both of these. This one's in the atria. This one's in the ventricles. Both of these work the same way. All right. So AMP and BMP, they work the same way. I have high blood pressure. It stretches my atria. My atria secrete atrial natriuretic peptide. Let's just go down. Let's go to the right first, then we'll go to the center, then we'll do the left. Atrial natriuretic peptide goes to the adrenal cortex. It turns off aldosterone release. Turns it off. It says no aldosterone, mister. So now aldosterone doesn't lead to sodium reabsorption, and water doesn't follow. So I have lower sodium reabsorption and less water that's reabsorbed. So it keeps my blood volume low and lowers my blood pressure. In addition, ANP goes to the hypothalamus and they turn off ADH release. ADH doesn't go to my kidneys and add the aquaporins, so I don't reabsorb water, so it keeps my blood volume low. All right. In addition, ANP goes to my juxtaglomerular apparatus of my kidneys, and it reduces renin release, which therefore reduces the formation of angiotensin 2, and it reduces vasoconstriction and actually can lead to vasodilation, and that lowers blood pressure. So ANP and BNP do the opposite of what you've been talking about, the renin, angiotensin, aldosterone system. It does the opposite of that. All right, so that's AMP. And this, I believe, is a collage. All right, let's go to the right first, like I just did last time. Oh, this is low blood pressure. This is what you're used to. This is not a collage. This is low blood pressure. You're used to this low blood pressure. That's the whole renin, angiotensin, aldosterone system and your autonomic nervous system. So you're used to this. All right, so what happens with low blood pressure? Well, low blood pressure leads to antidiuretic hormone release. Okay, it can do it. Remember, ADH also is called vasopressin. And remember, I told you the primary stimulus for release of ADH is a high osmolality. High osmo, and it's real sensitive to osmolality. But I did tell you that a low blood pressure directly will cause ADH release, but it has to be a drastic decrease in blood pressure. But it does directly do it. Well, ADH release increases water reabsorption. Meanwhile, remember the angiotensin aldosterone story. Angiotensin itself will cause increased ADH release, and you can see that right here, this line right here. Okay. My baroreceptors and my blood vessels detect low blood pressure. They turn on the sympathetic nervous system, which constrict my arterioles, and they raise blood pressure. This raises blood pressure. Okay? Blood pressure. In addition, I do the renin, angiotensin, aldosterone story. Renin converts angiotensin into angiotensin 1. ACE converts angiotensin 1 to angiotensin 2. Angiotensin 2 does a ton of things. First of all, it's a vasoconstrictor. You can see that right there. Secondly, it causes increase of aldosterone, which causes increased sodium reabsorption, which causes increased water reabsorption, which increases blood volume, which increases blood pressure. Okay, so we've done this 100 times. In addition, angiotensin 2 goes over to my brain. It makes me thirsty. I drink. It also increases ADH release, which makes me reabsorb the water I just drank. Well, it makes me reabsorb water out of my kidneys, so I don't urinate out the water I just drank. So that's with low blood pressure. The previous AMP was with high blood pressure. So you can see these regulatory mechanisms. And that's it for that part. Now we go into some other details of fluid, electrolyte, and acid-base balance.